Oh no, the AI sound. When two genetic loci produce identical phenotypes in cis and transposition, they are answer pseudoalleles. Explanation: when two genetic loci produce identical phenotypes in cis and transposition, they are pseudoalleles. The cis trans complementation test is done to determine if two mutations are allelic or non allelic by constructing a heterozygote. Pseudoalleles are the genes that functionally behave as allelic pairs, but structurally they are different genes present on two different genetic loci, not allelic pairs. Therefore, they behave as alleles in cis-trans tests and produce identical phenotypes. Multiple alleles refer to the presence of more than two alleles of a gene which occupy the same genetic locus. The part of the same gene occupy the same genetic locus and different genes occupy different genetic loci but do not produce an identical phenotype in cis-trans test. Amino acid substituted in sickle cell anemia is answer glutamic acid, for valine is beta chain. Explanation: amino acid substituted in sickle cell anemia is valine, for glutamic acid in beta chain. Sickle cell anemia is an autosomal disorder. The disease is due to the substitution of the 6 codon in the gene coding for the beta chain of hemoglobin. Due to this, the sixth amino acid in beta chain of hemoglobin, glutamic acid, is replaced by valine. Out of 80 GC pairing bases of DNA may exist in alternate valency state owing to arrangement called answer deuteromerization mutation. Explanation: out of 80 GC pairing bases of DNA may exist in alternate valency state owing to arrangement called deautonomerational mutation. Nitrogen bases are aromatic cyclic ketones with amine or imine group. Automers are a type of structural isomers which are formed by shifting of pi bond between functional groups. They are also called as ketoenol isomers and are interconvertible into each other as change to automerization converts the amino group of cytosine and adenine into amino group and keto to of thymine and guanine into enol group OH. This result in the pairing of atom automeric thymine with normal guanine and of cytosine with adenine. The resultant mutation is caused by autorizational mutations. Colostrum, the yellowish fluid secreted by mother during the initial days of lactation, is very essential to impart immunity to the newborn infants because it contains immunoglobulin A. Explanation: colostrum, the yellowish fluid secreted by mother during the initial days of lactation, is very essential to impart immunity to the newborn infants because it contains immunoglobulin A. During pregnancy, mammary glands of the female undergo differentiation, and at the end of pregnancy, milk secretion starts from them. This process is called lactation. The yellowish milk produced during the initial days of lactation is called colostrum. It is rich in carbohydrates, proteins, and antibodies and has very fat. Breastfeeding is very important for an infant because it provides them with nutrition and immunity. There are many types of antibodies produced in our body. Some of them are IgM, IgA, IgE, IgG. Normally, these antibodies are found in the blood and hence show humoral immune response. Colostrum contains a large amount of IgA, immunoglobulin A antibody, which provides immunity to the child and hence protects the child from many infections. In the immunity system, interferons are part of answer cytokine barriers. Explanation: the immune system interferons are a part of cytokine barriers. Interferons are members of a larger class of proteins called cytokine barriers, proteins that carry signals between cells. Most interferons are classified as alpha, beta, or gamma interferons depending on their molecular structure. Two other classes of interferon, omega and tau, have also been discovered. So far, more than 20 different kinds of interferon alpha have been discovered, but only beta and one gamma interferon has been identified. Malignant tertian subterranean fever is caused by Plasmodium falciparum. Explanation: malignant tertian subterranean fever is caused by Plasmodium falciparum. Malignant tertian Plasmodium falciparum in in which the cold stage is less pronounced and fever stage is more prolonged and intensified. If the fever is reducing, it occurs every 2 days. However, the fever is usually continuous or only briefly remitted. Plasmodium falciparum can cause severe malaria because it multiplies rapidly in the blood and can thus cause severe blood loss, anemia. In addition, the infected parasites can clog small blood vessels. When this occurs in the brain, cerebral malaria results a complication that can be fatal. 5 species of Plasmodium single cold parasites can infect humans and cause illness. Plasmodium falciparum or Plasmodium falciparum causes malignant tertian Plasmodium malariae or P. malariae. Humans occasionally become infected with Plasmodium species with a fever every third day. Plasmodium vivax or P. vivax with a fever every second day can remain dormant in the liver. Plasmodium ovale or P. ovale with a fever every second day can remain dormant in the liver. Plasmodium knowlesi or P. knowlesi, a fever pattern is daily. Genetic defect adenosine deaminase ADA deficiency may be cured permanently by introduction of genes isolated from the bone marrow cells which produce ADA into the cells of the patient at early embryonic stages. Explanation: the genetic defect adenosine aminase ADA deficiency may be cured permanently by introduction of genes isolated from the bone marrow cells which produce adenosine deaminase ADA into the cells of the patient at early embryonic stages. Adenosine deaminase ADA is an inherited disorder. It is a metabolic disorder. The mutation in adenosine deaminase ADA genes causes this disorder. It mainly affects the immunity system and causes severe combined immunodeficiency SCID. It was recognized as the first immunodeficiency or disorder. The the immune lacks the protection against bacteria, viruses, and fungi. This type of disorder is present in infancy, childhood, adolescence, or adulthood. The growth of the affected children is slower than the healthy children. The development is delayed in affected children. The adenosine deaminase enzyme is attached to adenosine deaminase ADA gene. If there is a decreased level of adenosine deaminase ADA in blood, then the person has adenosine deaminase ADA deficiency. Bone marrow transplant adenosine deaminase ADA enzyme is PEG vehicle can be used for the treatment of this disorder. The treatment adenosine deaminase ADA is a very complex process. Enzyme replacement therapy involves the injection of adenosine deaminase ADA to the patient. Introduction of genes isolated from bone marrow cells. It produces adenosine deaminase ADA into the cells of the patient at early embryonic stage. It can only cure permanently with the help of gene therapy. Main objective of production of herbicide resistant GM crops is to reduce herbicide accumulation in food articles for healthy safety. Explanation: main objective of production of herbicide resistant GM crops is to reduce herbicide accumulation in food articles for healthy safety. The pesticides which are used to destroy weeds are called herbicides. Herbicides are used in agriculture to control the weeds. They inhibit plant growth by blocking the biosynthesis of amino acids or photosynthetic activities of plants. Transgenic plants resistant to several herbicide example glyphosate, glufosinate, sulfonylureas, etc. have been successfully developed. The main objective of production/use of herbicide resistant genetically modified crops is to reduce herbicide accumulation in food articles for health safety. What would happen if a recombinant DNA is inserted within the coding sequence of an enzyme B galactosidase? Answer: this will result in the inactivation of the enzyme. The recombinant colonies do not produce any color. Explanation: blue white screening or insertional inactivation is a kind of method of selection and screening of transformed cells. In this method, the recombinants are differentiated from non recombinants on the basis of their ability to produce colored color in the presence of chromogenic substrate. The rDNA or recombinant DNA is inserted within the coding sequence of the enzyme B galactosidase. This results in inactivation of the enzyme in the presence of chromogenic substrate. Blue colored colonies are produced in the plasmid. In the bacteria doesn't have the insert, that is, those are non-recombinant. The presence of inserts in the bacteria recombinant result in the inactivation of B galactosidase enzyme, due to which colorless colonies are produced. The detritus food chain begins with dead organic matter. Explanation: the detritus food chain begins with dead organic matter. In detritus food chain, energy comes from organic matter or detritus generated in trophic levels of the grazing food chain. It begins with detritus such as dead bodies of animals or fallen leaves, which are then eaten by decomposers or detritivores. These detritivores are in turn consumed by their predators. It is usually smaller as compared to the grazing food chain. Primary consumers are detritivores, including protozoans, bacteria, fungi, etc., which feed upon the detritus saprophatically. In cockroach, the formation of ootheca is by the secretions of collateral glands of female. Explanation: collateral glands are present in female cockroach as part of reproductive system. These glands are branched tubular glands opening independently in the dorsal side of the genital chamber. The cell secretion produced by these glands forms the oothecal case of ootheca. Glomerulars occur in Bowman's capsule. Explanation: glomerules occurs in Bowman's capsule. Each kidney has nearly 1 million complex tubular structure called nephrons, which are functional units. Each nephron has two parts, the glomerulus and the renal tubule. The glomerulus is a tuft of capillaries formed by the efferent arteriole, a fine branch of the renal artery. Blood from the glomerulus is carried away by an efferent arteriole. The renal tubule begins with a double wrapped C-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus, along with Bowman's capsule, is called Malpighian body or renal corpuscle. Select the correct statement from the following. The ascending limb of the Henle's loop extends as the DCT. Explanation: a nephron is the structural and functional unit of kidneys. Broadly, Broadly, it has two parts: a glomerular capsule or Bowman's capsule and a tubule. A renal tubule has three main parts: proximal convoluted tubule (PCT), nephron loop, loop of Henle, and dis- distal convoluted tubule (DCT). The descending limb extends from the PCT and extend downwards into the medulla. The descending limb makes a hairpin turn in the medulla and extends upwards into the renal cortex. This part of the loop of Henle is called ascending limb of the loop of Henle as it ascends from the medulla to the renal cortex. The ascending limb of the nephron loop ends at the DCT. The DCT of several nephrons drains to a single collecting duct. The primitive prokaryotes responsible for the production of biogas from the dung of ruminant animals include the methanogens. Explanation: the primitive prokaryotes responsible for the production of biogas from the dung of ruminant animals include the methanogens. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. All methanogens share three common features. They are obligate methane producers, obtaining all or most of their energy for growth from producing large quantities of methane. They are Archaea belonging to the phylum Archaeota. They are strict anaerobes, limiting their growth to anaerobic environments. Which of the following is an innovative remedy for plastic waste? Burning in the absence of oxygen. Explanation: burning in the absence of oxygen is an innovative remedy for plastic waste. Pyrolysis is a thermochemical decomposition of organic material at high temperatures in the absence of oxygen. It involves the change of chemical composition as well as physical state. It is an irreversible process. The process is used heavily in the chemical industry to produce charcoal, activated carbon, to convert biomass into biochar, to turn waste into safely disposable substances. Cry2AB and Cry1AB produce toxins that control cotton bollworms and corn borer respectively receptively. Explanation: Cry2AB and Cry1AB produce toxins that controls cotton bollworms and corn borer receptively. A crop consisting of the cry gene shows resistance towards a group of insects. Cry protein is toxic in certain groups of insects. The toxicity is observed in the larvae of such insects. The protein which are encoded by Cry2AB control the cotton bollworms. The Cry1AB encodes a protein that controls corn borer. Corn Cotton bollworms are also controlled by proteins that are encoded by Cry1AC Examples of insects that are affected by cry genes are lepidopterans, tobacco birdworm, armyworm, coleopterian, Darius, that is beetles, and dipterans like flies, mosquitoes, etc. Which one of the following supports the multi-regional hypothesis of the origin of mod humans from archaic Homo sapiens? Answer: the existence of distinctive human races. Explanation: the existence of distinctive human races supports the multi regional hypothesis of the origin of mod humans from archaic Homo sapiens. Humans first evolved in Africa, and much of human evolved occurred evolution occurred on that continent. The fossils of early humans who lived between 6 and 2 billion years ago come entirely from Africa. Most scientists currently recognize some 15 to 20 different species of early humans. Which statement is incorrect regarding female external genitalia? Mons pubis is a cushion of fatty tissue covered by skin and pubic hairs. Explanation: Mons pubis is a cushion of fatty tissue covered by skin and pubic hair is incorrect regarding female external genitalia. The external genital structures of the female reproductive system are collectively termed as the vulva. The female external genitalia or vulva is the constitution of mons pubis, labia majora, labia minora, hymen, and clitoris. Mons pubis is defined as a cushion of fatty tissue covered by skin and pubic hair. The labia majora are particularly the fleshy folds of skin which extends down from mons pubis and surround the vaginal opening.